Hey guys, from Cell to Bond, we're excited to bring you guys a full AP Bio unit review video. This one video will cover all 8 units of AP Bio in under 30 minutes. To earn a good score on this test, make sure you guys are taking notes as you move along in the video. And if you need more practice, head over to our website and to the Quizlet we have set up to get the additional practice that you need. Let's jump over to Unit 1.
Hey guys, welcome to Unit 1, Chemistry of Life. This one's all about what life is actually made of and how those molecules work together to keep organisms alive. Alright, starting with water. So water is polar, which means its oxygen side pulls electrons harder than hydrogen does, giving a slight charge on both ends. That polarity lets water form hydrogen bonds, both with itself and with other polar molecules. These bonds are what make water so important for life, from keeping our bodies cool to letting plants pull water up through their roots. Water's got some super useful traits. So cohesion, water sticks to itself, helping it move up plant stems. Adhesion, it sticks to other surfaces like cell walls. High specific heat, it resists temperature changes, so it keeps our bodies and ecosystem stable. And surface tension plus evaporative cooling. That's why bugs can walk on water and sweat helps us cool off. Life is built from just 6 key elements, C, H, N, O, P, and S. Carbon is the backbone because it can make 4 bonds. Nitrogen and phosphorus are huge for building DNA and proteins, and sulfur helps shape some proteins too. Basically, these 6 elements are the building blocks of everything living. All big biological molecules or macromolecules are made of smaller pieces called monomers. You connect them by dehydration synthesis, which removes water, and break them apart by hydrolysis, which adds water. The 4 big macromolecule groups are carbohydrates, lipids, proteins, and nucleic acids. Carbs are made from carbon, hydrogen, and oxygen in a 1-to-1 ratio. Their monomers are simple sugars like glucose, and polymers are long chains like starch, glycogen, and cellulose. Their main jobs: quick energy, energy storage, and structure. Cellulose especially gives plant strength. Lipids are mostly nonpolar, which means they don't mix with water. They're built from glycerol and fatty acids, can be saturated, which are solid fats, or unsaturated, liquids like oils. They're used for long-term energy storage, building membranes, and making hormones. Phospholipids are special lipids with a water-loving head and water-hating tails. In water, they line up into a bilayer. That's the base of every cell membrane. It's selectively permeable, meaning some stuff moves through easily, and others things you transfer proteins to help. Proteins are chains of amino acids connected by peptide bonds. They do everything: act as enzymes, transporters, messengers, movers, and give structure. Shape equals function here. Even one amino acid change can mess up how a protein works. Protein shape has 4 levels. First, primary, the amino acid sequence. Secondary, local folds like alpha helices or beta sheets. Tertiary, the 3D shape formed by side chain interactions. And quaternary, which are multiple chains coming together like hemoglobin. Nucleic acids store and pass on genetic info. Each nucleoside has a sugar, phosphate, and a nitrogen base. DNA is a double-stranded with thymine. RNA is single-stranded with uracil. Base pairing goes A to T or A to U in RNA, and C to G. Those hydrogen bonds hold the strands together. DNA's 2 strands run in opposite directions. One, 5' to 3', and the other 3' to 5'. A double helix stays stable thanks to hydrogen bonds and complementary base pairing. The order of bases is what actually stores your genetic code for building proteins. And that's a chemistry of life. Everything living, from bacteria to humans, runs on these same molecules and reactions. If you don't want to practice, go to cell2bond.netlify.app. Today we're going to be doing AP Bio Unit 2, Cell Structure and Function Review. So, universal cell features. Every single cell on Earth, from bacteria on your sink to neurons in your brain, has 4 things minimum: a cell membrane keeping everything together, DNA for instructions, cytoplasm to hold it all, and ribosomes to build proteins. If a cell doesn't have these, it's not a cell. Prokaryotes versus eukaryotes. Prokaryotes are small, simple, no nucleus, DNA is free-floating, circular. So some examples are bacteria and archaea. Eukaryotes, they're larger and complex. They do have a nucleus, they have membrane-bound organelles, and they are plants, animals, fungi, and protists. Why cells stay? So small cells have a high surface to area to volume ratio. That means nutrients, oxygen, and waste moves quickly across the membrane. Big cells, the volume grows too fast, so diffusion gets slow and is bad for life. This is why organisms don't have one giant cell and they have 1 million small ones. Eukaryotes use membranes to create organelles so different reactions don't interfere. Some examples are lysosomes digesting junk, mitochondria making energy, or the ER folding and processing proteins. Mitochondria and chloroplasts used to be independent prokaryotes. A bigger cell engulfed them, which means they didn't digest them, and they became permanent roommates. So the evidence is they have their own DNA, the ribosomes, double membranes, and they replicate independently. So this will be on your FRQs. So the major organelles quick review. The nucleus is DNA storage and control center. Ribosomes build proteins. Rough ER folds and process proteins. Smooth ER makes lipids, detoxifies. Golgi packages and ships proteins. Mitochondria ATP powerhouse. Chloroplast for photosynthesis, and a vacuole for storage. Plants have a big central vacuole. The membrane is the phospholipid bilayer. So the fluid mosaic model, so you can see right here, contains a flexible and full of proteins, carbs, and cholesterol. It's selectively permeable, and stuff gets through easily and needs some help. So transporting across membranes, passive transport requires no energy. It's diffusion, facilitated diffusion, and osmosis. Facilitated diffusion uses channels, osmosis uses water. Active transport needs ATP, move things from low to high concentration. Some examples are the sodium potassium pump. So osmosis and water balance. Water always moves hypotonic to hypertonic, from low solid to high solid. Hypotonic means water enters, animal cells burst, and plants get turgid. Hypertonic means waters exit, cells shrink or wilt. Isotonic means balance, so water balance is equal to survival. So that's unit 2. Go to sodabond.netlify.app for 100 MCQ practice questions. So here's a few practice questions you can do. So today we're going to be reviewing Unit 3, Cellular Energetics. So, biological systems rely on energy and building materials to grow, reproduce, and maintain balance or homeostasis. In this review, we'll cover 5 key topics: enzymes, environmental effects on enzymes, cellular energy, photosynthesis, and cellular respiration. Each process connects to one main idea: how life transforms uses energy efficiently. Enzymes are biological catalysts, meaning they speed up chemical reactions by lowering activation energy. Their structure determines their function. The active site must perfectly fit into substrate like a lock and key. When they bind, they form an enzyme-substrate complex, which ensures reactions happen quickly and specifically. Enzymes are essential for regulating nearly every cellular process. So you can see the process right here. Environmental impacts on enzymes. Enzyme activity depends heavily on temperature, pH, and the surrounding environment. If conditions move too far from the optimum, enzymes can denature, losing their shape and, therefore, their function. Sometimes this can be reversed, but not always. Reaction rate also changes with substrate concentration. Too little and the enzyme isn't fully active. Too much and it becomes saturated. There's are also inhibitors. So there's 2 types of inhibitors. Competitive inhibitors block the active site. So like right here, you can see the inhibitor just like blocks it, right? And then non-competitive inhibitors bind elsewhere, changing the enzyme shapes through an allosteric effect. So right here, it isn't binding like to the active site right here. But once it binds to the bottom, it changes the shape, right? So it can't fit. So photosynthesis overview. Photosynthesis can be summarized with this equation: carbon dioxide plus water plus sunlight equals glucose plus oxygen. It occurs inside the chloroplast, specifically the thylakoids, where light reactions produce ATP and NADPH and the stroma, where the Calvin cycle uses that ATP and NADPH to build sugars. This process began in ancient prokaryotes and eventually created the oxygen-rich atmosphere that supports life today. So right here you can see a Calvin cycle. So photosynthesis light reactions. During the light reactions, photosynthesis systems 1 and 2 capture sunlight energy, exciting electrons. Water molecules split, producing O2, electrons, and protons. Those electrons move through an electron transport chain, creating a proton gradient across the thylakoid membrane. Then, ATP synthase uses that gradient to generate ATP. This process is called photophosphorylation. Both ATP and NADPH produced here go on to power the Calvin cycle and the stroma. Cellular respiration. So, cellular respiration converts energy stored in food into ATP. The molecule uses for energy. It happens mostly in the mitochondria and includes 3 main steps: glycolysis, which breaks down glucose into pyruvate, producing ATP and NADH. Krebs cycle produces pyruvate to form CO2, NADH, FADH, and ATP. And ETC use electrons to create a proton gradient, driving ATP production through oxidative phosphorylation. Finally, chemiosmosis occurs when protons flow through ATP synthase, forming large amounts of ATP. Aerobic is anaerobic energy. Aerobic respiration uses oxygen and produces a much higher ATP yield. Anaerobic respiration, or fermentation, happens without oxygen. It makes far less ATP and produces lactic acid or alcohol as byproducts. The cristae, or folds inside the mitochondria, increase surface area for ATP production. Some of the energy release becomes heat, helping endothermic organisms maintain body temperature. So here you see anaerobic, right? Here you see aerobic. So you see aerobic happens in the mitochondria. So the main idea: enzymes speed up life's chemical reactions. ATP is the main energy currency of the cell. Photosynthesis captures sunlight to make food. Cellular respiration breaks down that food to release energy. Together, these processes form the foundation of how life captures, stores, and uses energy to survive. So that's it. Make sure to look at the Quizlet card. Unit 4. It's all about how cells communicate and how the cell cycle keeps life going. By the end of this lesson, you'll understand why cells talk to each other, how they send messages, and what happens when things go wrong. So here are 3 main takeaways. Number 1, cells communicate using chemical signals. Number 2, feedback helps keep balance. That's homeostasis. And number 3, the cell cycle keeps life continuous by making sure cells grow and divide properly. Cells don't just flow around doing random stuff. They talk to each other to coordinate things like growth, defense, and repair. They send off chemical messages like hormones or electrical messages like nerve impulses. This communication keeps multicellular organisms like us organized and balanced. There are 3 types of cell signaling. First is direct contact, where cells literally touch like immune cells or through tiny channels called plasmodesmata in plants. Second, local signaling, short distance communication like neurotransmitters between nerve cells. And third, long distance signaling, which uses hormones that travel through the bloodstream. When a signal reaches a cell, it goes through 3 main steps: reception, transduction, and response. Reception means the signal, called a ligand, bonds to a receptor. Transduction is when the message is relayed inside the cell through a series of proteins, and response is when the cell actually does something like make the protein or device. Think of receptors as locks and ligands as keys. When the right key fits into the lock, the receptor changes shape, and that shape change starts the communication process. Receptors can be found on the cell membrane or inside the cell, depending on the type of signal. The message inside the cell gets passed along like a relay race through different proteins. This involves phosphorylation cascades, basically adding phosphate groups to activate or deactivate proteins. There are also second messages like Ca and P, or calcium ions, which help amplify the signal and make sure the message is loud and clear. After the message is passed along, the cell responds. This means turning genes on or off, making new proteins or enzymes, secreting molecules, or even triggering cell division or apoptosis, which is programmed cell death. Basically, the signal leads to some kind of action. Here are a few examples. Cytokines trigger immune cell division. Yeast pheromones start mating behavior. Epinephrine, also called adrenaline, makes liver cells release glucose. Ethylene ripens fruit, and the SRY gene starts neural development in embryos. Cells also change their responses depending on what's going on around them. Bacteria use something called quorum sensing to detect population size. Plants respond to light hormones, and animals respond to stress or temperature changes. So environment plays a big role in how signaling works. Feedback loops help the body maintain homeostasis. Negative feedback brings the system back to normal, like how sweating cools you down when you're hot. Positive feedback pushes the system further until something's complete like childbirth or blood clotting. These loops are what keep our internal system stable. Now let's talk about cell cycle. Basically how cells grow and divide. It goes like this. G1 is where the cell grows. S phase is where DNA replicates. G2 is prep time for division. N phase is mitosis, where the nucleus divides, followed by cytokinesis, where the cytoplasm splits, and some cells are just shown G0, which is a resting phase. Mitosis is how cells make exact copies of themselves. There are four main steps. Prophase, chromosomes form. Metaphase, they line up. Anaphase, they separate. And telophase, new nuclei form. After that, cytokinesis splits the cells into two identical daughter cells. The cell cycle is tightly controlled by checkpoints at G1, G2, and M. Cyclins and CDKs act like traffic lights, telling the cell when to go or stop. If these checkpoints fail, cells can divide uncontrollably, which can lead to cancer. Sometimes the system triggers apoptosis, self-destruction to prevent damage. To wrap up, cells communicate through signals, coordinate activity. Signal pathways lead to cell responses like growth or gene changes. Feedback loops maintain internal balance, and the cell cycle controls growth and repair. If something goes wrong, that's when diseases like cancer can develop. That's it for Unit 4. If you want to practice what you learn, check out Cell to Bond on Netlify. It's all activities and review. Questions to have everything sick. Thanks for watching. Unit 5. Heredity is one of the biggest units on the AP Bio exam. It contains genes, meiosis, Punnett squares, and all of that. So what is meiosis? Meiosis is a process that produces haploid gametes, reduces chromosome number, passes DNA to the next generation, and creates genetic variation. The end result is 4 genetically unique haplocells. So here's a quick rundown. So in prophase 2, the spindle forms, nuclear envelope breaks, and chromatids are still connected. Metaphase 2, chromosomes line up in a single file. Anaphase 2, sister chromatids separate for the first time. Each becomes an individual chromosome. Telophase 2, nuclei reform, cytokinesis, and you end up with 4 unique cells. So, mitosis versus meiosis. Mitosis is 2 identical diploid cells, no crossing over, for growth or repair. Meiosis is 4 unique haploid cells. Crossing over happens and it's for sexual reproduction. Both use spindle fibers, but their outcomes are totally different. So genetic diversity in meiosis, there's 3 main sources. So there's crossing over in prophase 1, independent assortment, and random fertilization. So here's crossing over. Non-sister chromatids swap DNA, creates recombinant chromosomes. It happens in the chiasmata. So there's more sources here: independent assortment, random fertilization. So genotypes, phenotypes, and crosses. So genotype is an allele combination like BP, RR, or YY. Phenotype is a physical expression. Punnett squares predict probability, not certainty. So you should know monohybrid crosses, dihybrid crosses, and test crosses. So sex-linked traits. Traits on the X or Y chromosome. X-linked traits are more common in males, XY individuals, because they only have one X. Some examples are colorblindness or hemophilia. And there's non-nuclear inheritance. Okay, so here, some environmental factors can affect gene expression. So same genotype is different phenotypes depending on temperature, nutrition, light, and stress. Some examples are temperature-dependent sex in reptiles. So for the previous one here, we had non-nuclear inheritance. So the mitochondria and chloroplast genes are maternally inherited. So you always get mitochondrial DNA from your mom, but this does not follow Mendelian ratios. So that's AP Bio Unit 5. So everything you need to know for the exam. So here's are some practice questions. There's another one. Some more. So go to cell2bond.netlify.app for 100 MCQ. Today we're going to be talking about Unit 6, Gene Expression and Regulation. So here's the criteria for genetic material. Store information, instructions for development and metabolism, stable and replicable. They need accurately copied during cell division and undergo changes. They need to be capable of mutations for diversity. Here's the big debate in Griffith's experiment. So the debate in the 1920s was proteins had 20 amino acids which made them more complex, which leads to likely genetic material. Nucleic acids, there's only 4 nucleotides. It seems too simple. So Griffith's experiment in 1928 had a discovery that we found a transforming principle that changed harmless R bacteria into deadly S bacteria. The conclusion was genetic information could be transferred between bacteria. So here are three people. These people showed destroying DNA stopped transformation, proving DNA carries traits. Percy Chase used radioactive labels showing DNA enters bacteria, confirming DNA as genetic material. Key discoveries for DNA structure. So here are Chargaff's rules. A equals T and C equals G. The ratios are constant across all species. Rosalind Franklin and Maurice Wilkins used X-ray diffraction. They revealed DNA as a helix with a consistent diameter. Watson and Crick in 1953 used Chargaff's rules and Franklin's image to build the first accurate 3D model of the double helix. DNA and RNA structure basics. So the base pairing is A to T, there's 2 hydrogen bonds, and C to G, 3 hydrogen bonds. These hydrogen bonds hold the strands together, and phosphodiester bonds is part of the sugar phosphate backbone. So DNA replication models. From the Nielsen and Stahl experiment, they used heavy and light nitrogen isotopes. The result after one round was hybrid DNA, with one heavy and one light strand. The conclusion they got was they supported the semi-conservative model, which each new DNA has one old strand and one new strand. Steps in enzyme DNA replication. There's unwinding, where the helicase breaks hydrogen bonds, SSB proteins stabilize strands, relieving tension, topoisomerase prevents overwinding ahead of the fork, embedding new strands, DNA polymerase adds complementary nucleotides to each template strand. The result is 2 identical DNA molecules. The central dogma of transcription. So the central dogma of biology is DNA turns to RNA, which goes to the proteins. This transcription is how you go from DNA to RNA. It happens in nucleus and eukaryotes. The process is RNA polymerase uses one DNA strand to build complementary RNA. The key RNA types are mRNA, messenger, rRNA, ribosome, and tRNA, transfer. Base pairing RNA is as goes. DNA A bonds to RNA U, DNA T bonds to RNA A, and DNA G/C bonds to RNA C/G. RNA processing eukaryotes. The initial product is pre-mRNA, which has introns and exons. Modifications before leaving the nucleus are the 5' cap modified, GTP added, poly-A tail, around 200 adenine nucleotides added to the 3 end. RNA splicing. The introns are removed and the exons are joined together. The result is mature mRNA ready for translation. In translation, it happens in the ribosome, in cytoplasm, or on the rough endoplasmic reticulum. The process as goes. First is initiation. Ribosome binds to mRNA as start codon, OG. tRNA with UAC anticodon brings methionine. Elongation. Ribosome reads codons. tRNAs bring matching amino acids, peptide bombs home. And termination. Ribosome reaches a stop codon. Here are 3 of them, UAA, UAG, and UGA, and polypeptide chains released. Gene expression regulation. So why should they regulate? Cell differentiation, timing, and response to environment. Key regulatory sequences. The promoters with RNA polymerase lines. Operators, the on and off switch for transcription. The enhancer increases transcription rate, and the operons, prokaryotes, are groups of genes controlled together like a lag operon. Epigenetics. Epigenetics is the heritable changes in gene expression that do not change the DNA sequence. The mechanisms are high stone acetylation, as acetyl groups loosens DNA, increases transcription. DNA methylation adds methyl groups to DNA, which tightens the chromatin and silences transcription. Mutations. So the causes are random errors during DNA replication repair, mutagens, radiation from like UV or X-rays, chemicals like smoke, and infectious agents like HPV. Types of gene mutations. There's substitution, where one base is changed, insertion/deletion, which is frame shift, adds or removes bases, shifting the reading frame. Biotechnology techniques. There's gel electrophoresis, which separates DNA fragments by size, the smallest moves furthest. PCR, polymerase chain reaction. It makes millions of copies of a specific DNA sequence. DNA sequencing determines the exact order of nucleotides, and bacterial transformation, inserting foreign DNA into bacteria using plasmids and restriction enzymes. So go to solderbond.lfi.app for 100 MCQ practice questions, and that'll be all. Thank you. Today we're going to be covering Unit 7, Natural Selection. So evolution by natural selection. The key question is, how does the population's genetic makeup change over time? So Charles Darwin in the Galapagos Islands, he studied finches, and he found that finches' beak shapes varied based on food source. He also studied tortoises, which shell shapes varied based on diet. So evolution, he called it descent with modification, which is the change in genetic makeup of a population over time. Key observations of Darwin was heritability. Traits are passed from parents to offsprings. Adaptations, inherited traits that enhance survival and reproduction. Some key concepts are fitness, which is the ability to survive and reproduce, which is measured by reproductive success, selective pressure, and environmental changes that make certain traits advantageous. The result is favorable traits accumulate in a population over generations. Populations evolve, not individuals. Some examples of natural selection, like the peppered moss. In the Industrial Revolution, the suet on trees led to dark moss becoming more common. Antibiotic resistance, so bacteria survive drugs, which leads to resistant genes increasing frequency. And pesticide resistance, insects survive sprays, so resistant populations evolve, like DDT. Key point is the overproduction of offspring leads to competition, which leads to differential survival of the fittest. Evolution basics, so evolution means descent with modification. Populations change genetically over time. Fossils support evolutionary history. DNA comparisons show relationships, and multiple scientific fields confirm evolution. Artificial selection. Both lead to evolutionary change. So natural selection, nature selects for survival and reproduction, so like camouflage and disease resistance. In artificial selection, humans select for desired traits. So dog breeds, crop plants, domestic animals, usually for more yield, taste, or appearance. Population genetics and gene control. The genetic makeup of populations. Population is a group of same species in the same area interbreeding. The gene pool is the total genetic diversity of all alleles in a population. So here's how genetic composition changes. 1. Through mutations, which are random DNA changes which lead to new alleles, source of variation. Prokaryotes mutate faster because they have short generation time, and eukaryotes mutate slower because they have longer generation time. Genetic drift, there's two types, bottleneck and founder effect. So for the bottleneck effect, it's when disaster drastically reduces population size, like the black robin. It reduces genetic diversity. The founder effect is when a small group isolates to form a new population, like the Amish population. It's reduced genetic diversity and unique allele frequencies. Genetic drift is when random changes in allele frequencies, especially strong in small populations. Gene flow and modes of selection. Migration, how selection shapes traits. Gene flow is movement of alleles between populations, which increases diversity. You also have modes of natural selection. Directional favors one extreme, shifts the mean. Is usually homozygous recessive. Stabilizing favors the middle, so it doesn't favor any extreme. It reduces variation. And disruptive favors both extremes, which increases variation and can lead to new species. Here's evidence for evolution. First, we have the fossil record. It shows organisms from the past differ from today. It shows the timeline of change. Second is geographical evidence. Closely located species more similar, even if environments are similar elsewhere. 3. Geological evidence. Rock layers show older fossils in lower layers. Phylogeny and cladograms showing evolutionary relationships. The node, which is the branch point, speciation event, it shows how common ancestors diverge. The outgroup is the least closely related species, which is used for comparison. Shared derived traits is when traits that indicate common ancestry. So a phylogenetic tree shows time is calibrated with fossils or molecular clocks. The branches indicate when divergence occur. If for a cladogram, there's no time scale, it only shows relationships. The branches only show order of divergence. Speciation. So this is how new species form. The biological species concept is a population that can interbreed to produce viable and fertile offspring. Reproductive isolation. So there are barriers to interbreeding. Pre-zygotic, which is before fertilization, or have different mating seasons, behaviors, or cause. Post-zygotic, after fertilization, is hybrid species are sterile, like mules. Allopatric versus sympatric speciation. So it's geographic versus same area speciation. For allopatric, it's physical or a geographic barrier like a mountain or river. The population is separated, which leads to them evolving independently. For sympatric speciation, there's no physical barrier. Reproductive isolation occurs within the same area. This causes differences in mating preference or ecological niches. Speciation rates, there's gradualism, which is slow, steady accumulation of small changes, and punctuated equilibrium, which is a rapid burst of change followed by long stability. Types of evolution extension, there's divergent, convergent, and extinction. So divergent evolution is when a common ancestor leads to a different species which have different traits, like Darwin's finches. Convergent evolution is when different ancestors lead to similar traits, same function, so like bat wings and bird wings. And it has analogous structures. Extinction, mass extinction, at least 5 major events, so like the dinosaurs. Current concern is that human activity may be causing a sixth mass extinction. Origin of life, Miller-Urey experiment. How did organic molecules form? Here's the timeline. Earth formed 4.6 billion years ago. The life-supporting environment happened 3.9 billion years ago. The earliest fossils we have are 3.5 billion years ago. So the Miller-Urey experiment in the 1950s, the setup was simulated early Earth, water, ammonia, methane, hydrogen, and electric sparks for lightning. The result was it produced amino acids, building blocks protein. The significance was that organic molecules can form spontaneously under Earth's early conditions. So the Unit 7 summary, here are some key takeaways. 1. Natural selection. Favorable traits accumulate in a population due to differential survival and reproduction. 2. Genetic drift. Random changes like the bottleneck or the founder effect affect small populations strongly. 3. Gene flow. Movement of alleles between populations increases diversity. 4. Evidence for evolution. There's fossil record, homologous structures, DNA comparisons, and embryology. 5. Speciation. New species formed via reproductive isolation, allopatric or sympatric. 6. Miller-Urey. He demonstrated organic molecules can form spontaneously on early Earth. So go to solderbond.lfi.app for 100 MCQ practice questions. Thank you. Today we're going to be covering Unit 8, Ecology, Interactions with Organisms and the Environment. So the definition of ecology is the study of interactions between organisms and their environment. There's levels of study, from smallest to largest. It goes from atom, molecule, to organelle, to cell, tissue, to organ system, organism, population, ecosystem, biome, and biosphere. So abiotic versus biotic factors and responses. Abiotic factors are non-living, things like temperature, water, sunlight, wind, soil, minerals. Biotic factors are living, plants, animals, bacteria, fungi, and protists. Some organism responses are behavioral, like phototropism, which is plants bending to light, or fight or flight. Physiological, internal process changes like heart rate or hormone release. Animal communication, so information exchange and fitness. Organisms communicate to influence behavior and increase fitness. So here are some, like mating signals. Some examples include fireflies glowing, peacock tails. Some like defense, things like cobra hood, skunk spray. For social bonds are baboons grooming, dogs licking pups, and territory is like cat scent marking or ants using pheromone traits. So the key point is that behaviors that increase survival and reproduction are favored by natural selection. Symbiosis. Symbiosis is any close long-term relationship between species. So there's three types. Mutualism is where both species benefit, so like bees and flowers, nectar for pollination. Commensalism is when one benefits and the other is unaffected, so like barnacles on whales. And the third one is parasitism, where the parasite benefits but the host is harmed, like ticks on dogs. Energy strategies and metabolism. So thermoregulation and reproduction. Thermoregulation has endotherms, which generate internal heat, like mammals or birds, or ectotherms. Body temperature matches the environment, like reptiles and amphibians. There's a few reproductive strategies. Long day breeders mate in summer, longer days. Short day breeders mate in winter, they give birth in summer. The metabolic rate is larger body mass, which equals a lower metabolic rate per kilogram, like elephant versus the mouse. Trophic levels. There's 5 trophic levels. So we have primary producers, autotrophs. They make their own food using lighter chemicals like plants, algae, chemosynthetic bacteria. Primary consumers, the herbivores, they eat producers like rabbits, grasshoppers. Secondary consumers, the carnivores, they eat herbivores like snakes or foxes. Tertiary consumers, they eat other carnivores like hawks, orcas. And decomposers break down dead material, they recycle nutrients. So here are like fungi, bacteria. Energy flow through ecosystems. Food chains and food webs. Energy cannot be recycled, unlike matter. The sun continuously supplies energy. The food chain is a linear energy transfer. It goes from producer, primary consumer, secondary consumer, tertiary consumer. The food web is interconnected network of food chains. The impact of energy changes, so more energy leads to more or bigger trophic levels. Less energy leads to fewer or smaller trophic levels. Population growth models. There's exponential versus logistic growth. Exponential growth is under ideal conditions, unlimited resources. It has a J-shaped curve, growth rate constant, which R max. The formula is dn divided by dt, which is R max times n. Logistic growth is realistic conditions, limited resources. It has an S-shaped curve. Growth slows near carrying capacity, which is k. So the formula is d of n divided by dt equals R max times n times k minus n all over k. So the carrying capacity k is maximum population size an environment can support. Community ecology and species diversity. Measuring diversity. There's a community of populations of different species living together and interacting. Community structure equals species composition, which species, plus species diversity, how many and how equal. So Simpson's diversity index accounts for both species richness, number of species, and species evenness, relative abundance. Higher diversity leads to a more resilient ecosystem. Interspecific interaction. So there's competition and predation. Competition is different species compete for same resources. The outcome is competitive exclusion. Inferior species are eliminated locally. Niche partitioning. Species divide resources to avoid competition. It allows coexistence to realize niche. Predation. Predator kills and eats prey. It's a plus minus relationship. Trophic cascade. Predators control prey density, affecting lower trophic levels. The outcome is indirect control of ecosystem. Keystone species and disturbances. Ecosystem stability and change. Keystone species are not usually abundant, but other species rely on them. Remove them cause devastating effects. For example, sea otters, which control sea urchins and protect health forests. So disturbances are events that remove organisms or resources, like fires, floods, human activity. Biodiversity. More diverse ecosystem leads to more resistant to change. Invasive species and human impact. So there's some threats to biodiversity. Invasive species established outside of native range, usually via human activity, grow and reproduce quickly, outcompete native species. For example, insects and imported produce crates. Major threats from human activity like habitat loss and deforestation. Invasive species outcompeting natives. Overharvesting. Faster than populations can rebound. So eutrophication and biomagnification. Pollution and toxin accumulation. So the issue here, eutrophication, is when excess nitrogen runoff leads to algal blooms. It depletes oxygen and suffocates fish. This could happen through fertilizer runoff. So biological magnification is when toxins become more concentrated at higher trophic levels, like mercury in fish to humans to eat fish, and this can happen through normal pollution. So to visualize biomagnification, think of a small fish which has low toxin. It gets eaten by a big fish who has more toxin. And then the human eats big fish and it has the highest toxin. Global change, climate and atmospheric alterations. Global change alters the climate and atmospheric chemistry and ecological systems that reduce Earth's capacity to sustain life. Some examples are air and water pollution from industrial and agricultural contaminants, acid rain from air pollution which harm ecosystems, CO2 emissions contribute to global warming, ocean acidification is when increased CO2 absorbed by oceans affects marine life, like shellfish and coral. So summary is ecology is the study of interactions between organisms and their environment, abiotic and biotic factors. Energy flows one way through trophic levels. It cannot be recycled. Population growth is exponential, J curve, in ideal conditions, and logistic, S curve, near carrying capacity. Community interactions include competition, predation, mutualism, commensalism, and parasitism. Biodiversity increases ecosystem resilience. Human activities lead to habitat loss, invasive species, overharvesting, pollution, and climate change. These are major threats to the ecosystem. And go to Stutterbond.nullify the app for 100 MCQ practice questions.